Good morning, everybody. Today we're going to do a quick review about enzymes, and then we're going to look at some of the enzyme patterns and trends that we have been covering in the past couple days. So we have previously taken notes about enzymes and their structure, what type of macromolecule they are, and what their overall purpose is. And the past couple days, we've been looking at specifically the enzyme known as catalase, found in chicken liver, which breaks down hydrogen peroxide. And through that, we've seen a couple of trends and patterns. So we're going to start with a review of what an enzyme is, and then we'll look at those trends and patterns in the second half of this. So to start off our review, what is an enzyme? We've already covered that an enzyme is a type of protein. A reminder, a protein is made of a monomer known as amino acids. Enzymes specifically act as catalysts. They have the ability to speed up how quickly a reaction is going to happen by lowering the overall activation energy. So they decrease how much energy is needed to start that reaction. Looking at the picture on the right, we've seen before, the top picture is showing us how much energy it would take without the presence of an enzyme to break lactose into glucose and galactose. We can see just how tall that bump is. That means there's going to be a lot of activation energy needed to start that reaction. In the bottom picture, we see lactase. Lactase is an enzyme. It ends in ASE. That's the hint of what an enzyme is typically named as. With the presence of lactase, lactose can be broken down with less activation energy. So we see how much shorter that bump is on the graph. That means less energy is needed to start the reaction, and the lactose is broken down into glucose and galactose faster. The enzyme has a specific shape to it. Notice how I'm saying enzyme has an active site. That means that the enzyme has a specific smaller part of it known as the active site. The active site is always on the enzyme. That active site fits one specific substrate. We talked about how this is like a lock and a key. The enzyme is the lock. The key is the substrate. If the two pieces fit together, the reaction can occur. If the two pieces don't fit together, the reaction cannot occur. So the enzyme can convert that substrate into products. Looking at the picture to the right, we can see that. The dark blue shows us the enzyme. The light blue shows us the active site of the enzyme. The green is the substrate. It locks into that active site and is broken into red and purple, two products. We've also talked about how some enzymes can break down a substrate into two or more smaller parts, whereas other enzymes have the job of joining multiple substrates together to make one final product. So I just want to remind you, even though most of the enzymes we've seen so far have been breaking apart a substrate, there's other enzymes that do the opposite and join together multiple smaller parts. We then also looked at some examples of enzymes. This is a very short list. I don't expect you to know these. There's actually thousands and thousands of enzymes inside almost every cell in our bodies. Each enzyme breaks down something different. So we need a whole bunch of different enzymes to break down or join together a whole bunch of substrates. We just talked about how there's a few examples here that we might be familiar with, such as lactase, maltase, amylase, protease, sucrase, and we talked about how this pattern of ending in ASE. That's just a good hint for us to remember if we see something ending in ASE, it's likely one of those enzymes. What we're going to look over now is how some four factors can impact the reaction rate of enzymes. You have already looked over this in your pair deck from Monday and Tuesday. What we're going to do now is just kind of solidify it. You discovered this on your own. Now let's talk about it, finalize it, and make sure we are all understanding what the main idea is. So the four trends we're going to be looking at. First up, how does enzyme concentration impact the rate of a reaction? Second, how does substrate concentration impact the rate of reaction? How does pH impact rate of reaction? And lastly, how does temperature impact the rate of reaction? So just a couple reminders here. Rate of reaction or reaction rate, those are synonyms, and it's talking about how fast does a reaction happen. The faster the rate, the faster it's going to happen. The slower the rate, the longer it's going to take. So let's go ahead and look at this first trend. This first trend is looking at how enzyme concentration impacts the rate of the reaction. We've already seen this slide before, but it's showing us the enzyme in green and the substrates in blue triangles. On the left, we have one cell. We have one cell that has one enzyme and multiple substrates. Now, if we take another cell and we put it on the right, and it has 5 enzymes and an equal number of substrates. We can predict which of these will have a faster reaction rate. Like we talked about in class, the reaction rate will be faster on the right because there are more enzymes. So I'm going to go ahead and just close out my camera so you can see the full screen right now. But we can see that main conclusion here. The substrates will be changed into products faster on the right because there are more enzymes present to catalyze that reaction. What that means is that as enzyme concentration increases, the rate of the reaction increases. It becomes faster. We can represent this graphically. So in assignment 50 later, we're looking at a couple different graphs and how to interpret them. Looking at the graph here, we can see that we have rate of reaction on our y-axis. Every single graph is always going to have that on the y. Rate of reaction, reaction rate on that y-axis. Then the bottom, we have enzyme concentration. As enzyme concentration increases, the rate of the reaction increases. That is what we saw on the previous slide. The more enzymes there are, the faster the reaction will happen because there's more enzymes available to bind to the substrates. So this is a pretty short and simple trend that we can notice. Now let's go ahead and look at our next trend. How does substrate concentration impact the rate of the reaction? Reminder, the substrate in this picture is being shown in those blue triangles. On the left right now, we have a low concentration of substrate. Now let's say that on the right, we keep the same number of enzymes, but we increase the number of substrates. That would look something like this, where again, still only 3 enzymes, but additional substrates have been added. Take a moment, think to yourself, pause if you need to. What do you think would happen to the reaction rate if we kept adding more and more substrates? The answer is that the substrates or the enzymes would be at the same reaction rate on both sides. Go ahead and, sorry, that's one typo on my slides today. The enzymes would have the same reaction rate on both sides. No matter how much I increase the substrate concentration on the right, it would not change the speed of the reaction. Since the enzyme concentration is equal on both sides, the reaction could not speed up any faster. So this shows the second trend. The substrate concentration reaches a point of saturation where the rate stays constant. Graphically, we can see that in this picture. This picture is now showing us the rate of reaction on the y-axis and the substrate concentration on the x-axis. We can see how this graph starts to raise up, but then eventually levels off. Since it's leveling off, that's saying the reaction rate stays the same. At this point in the graph, the enzymes are saturated. Adding more substrates won't make them go any faster. All the enzymes have a substrate each. They can only do that reaction so quickly, the rate stays the same. So I summarized this on the left in 2 bullet points. The first one describes the first part of the graph where it's increasing. And it says increasing the substrate concentration will initially increase the reaction rate until all of the available enzymes are used up. Once all of the available enzymes have a substrate, the reaction rate will slow. Second bullet point, once all of the available enzyme active sites are occupied, have a substrate bound to them, increasing the substrate concentration further will have no effect on the reaction rate. So if I go back to my previous picture, on the previous slide, no matter how many triangles I put inside the right side of the cell, no matter how many substrates I added, the rate would stay the same. I could add 2 million, the rate would not change because there are only 3 enzymes. So with that, we've now covered 2 trends that impact the reaction rate of an enzyme without changing the enzyme structure. So those first 2 trends are looking at what happens if I change how many substrates. What happens if I change how many enzymes? The next 2 trends we are going to look at are now going to potentially impact what the enzyme active site is like. With a change to the enzyme's active site, it can lead to a change in the reaction rate. So yesterday in your pear deck, you looked at what denaturation means. These 2 trends of pH and temperature can lead to denaturation. So let's look at this first one, pH. pH has an ability to slow or speed up a reaction rate. When the pH is optimal, the rate of the reaction will be the fastest. That's when it's the best time for that enzyme. It's perfect conditions. That's a synonym for optimal, perfect, best, amazing. So in this first example, the optimal pH for pepsin, the highest point on this graph corresponds to the highest reaction rate. So this y-axis says rate of reaction backwards for some reason, but we can see how this high point corresponds to a pH of 2. That means that will be the optimal pH for pepsin versus the optimal pH for trypsin, found in a different part of the body, actually has an ideal optimal pH of 8. When the pH is non-optimal or extreme, the reaction rate will either slow or stop. So we can see that as we move down, the rate of reaction gets slower or stops. If I took the enzyme pepsin and I put it into a pH of 5, the graph does not extend to 5. That means pepsin is not able to do any reactions in a pH of 5. If I compare that to a pH of 3, it could still do a couple reactions, but it would be a lot slower. Since the rate is lower on the y-axis, that means it will be slower. So why is it that it slows down and stops? The non-optimal or extreme pH can slow the reaction rate. The enzyme can become that word of denatured by non-optimal or extreme pH. When it's in its optimum, the active site fits the substrate. When it's in the incorrect pH, the active site actually changes and is no longer able to bind to the substrate. We can also see this in temperature. Temperature can slow or speed up the reaction rate as well. When the temperature is optimal, the reaction rate will be the fastest, represented by the highest point in the graph. So in humans, it's typically around 40 degrees Celsius looking at this graph. If it goes too high or it goes too low, the reaction rate will slow. In other organisms, the temperature might be different. So hot springs bacterium that live in 150 degrees Celsius bodies of water survive at about 70 degrees Celsius for the enzyme. Same idea here. Why does the non-optimal or extreme temperature slow the rate of reaction? If the heat is too high or the heat is too low, the active site of the enzyme can change. With a change in the active site of the enzyme, it can no longer bind to the substrate. The enzyme has been denatured and the reaction cannot be completed. We can see that in this picture we've seen before. On the left, we have the enzyme in optimal conditions where it has the correct shape of the active site, versus on the right, if the temperature is too high or too low, or the pH is too high or too low, the active site has changed shapes. that means that their action would be faster on the left because it is in optimal conditions and it is able to bind to that substrate. So these two ideas, non-optimal conditions, can be created by incorrect pH or incorrect temperature. This is a quick little summary slide. These are the 4 trends in the rate of reactions or the reaction rates, and what we covered just now. How does enzyme concentration impact the rate of reaction? Increasing enzyme concentration increases the reaction rate. How does substrate concentration impact the rate of reaction? Increasing the substrate concentration will increase the reaction rate until all the enzymes are used up, at which point increasing the substrate concentration will have no impact on the reaction rate. That's that flat line on the graph. 3 and 4, how does pH and temperature impact reaction rate? Both pH and temperature can increase or can decrease the reaction rate. What you should be able to do now is look at a couple of graphs and interpret them for what are the optimal conditions. And that is what we are going to do on assignment 50. So assignment 50 is based on what we just talked about now and what we worked on Monday and Tuesday. It is a Google form. It is not a quiz though, so don't stress right now. Take the stress away. It's just an assessment of where are you at. Since it's not a quiz, if you are unhappy with your score, it will automatically tell you which ones you missed and you can do a retake. So you can go ahead and now close out of this video and complete assignment 50.